Hello everyone, my name is Andreas Dräger and I like to welcome you to this lecture. I'm the junior professor for computational systems biology and I started my work at the University of Tübingen in July 2018. And today I have the pleasure to introduce you to systems biology, which is the second part of this lecture. So this means we're moving now from the structure part to a new topic. And therefore, we're starting over. And I first want to give you a brief introduction what systems biology is in general. And then we will give a more broad overview about different topics that are of interest. So first of all, when you look at cellular systems, you are confronted with an enormous complexity. Here you see a very simple cell, a bacterium, which is E. coli, drawn by David Goodsell in his book, The Machinery of Life. And that's a picture that is drawn to scale. And you see a lot of details here. And what you notice is that already such simple organisms are very complex. They're densely packed with numerous different molecules in a very crowded environment. And so you can see that in this very small scale that we are looking at, there are different rules apply because here things are different from a beaker in the sense that molecules don't flow around in a well-stirred, a well-mixed, homogeneous fluent. Instead, there are many obstacles and everything is highly organized. And so far, in the first part of this lecture, we covered the structure bioinformatics, where we thought about individual molecules, atoms, and bonds, and so forth. Now we're looking at the interactions between those different items and how they interplay in a very complex scenario. And on the left-hand side, you see that there is a lot of interest in that. So first of all, metabolism is not a boring science. It's really worthwhile to look at. For instance, the motivation is that if we understand metabolism well enough, we might be able to engineer cells to produce what we desire to have. For instance, very high amounts of substances that are of interest, or even solving our energy problems, and so forth. So this slide gives you an overview or a definition of systems biology based on what is written in Wikipedia. And Wikipedia brings it to the point, and that's why we're looking at that definition here. So systems biology is really the systemic view driven by mathematics. So we're introducing mathematics to biology and try to establish a theoretical foundation for biology and how cellular systems work, including their complex interactions and how everything works together. So, and this is in contrast to the reductionist view that is the traditional biology, and we will get to that in a minute. So here we don't look at individual items, but at the system as a whole, and this brings us to new functions, to new properties of the overall system, in contrast to the properties of individual components. And therefore, we can study the whole system under a different point of view, from a different angle, and think about it very differently. And that happens on different levels: cells, tissues, or entire organisms, or interaction of organisms, and tries to describe everything on a mathematical basis so that we can come up with general principles and describe what happens mathematically. So I mentioned that already. Reductionism, what is it? That's the traditional approach to biology. A reductionist reduces a complex system to the most atomic part. So basically you can think about decomposing the system until you have individual items that can't be further decomposed, and then you study the properties of those entities. So, and I mentioned it's the traditional approach to biology, because what reductionists do is basically generating long lists of things, such as collecting information about all organisms on Earth. So what organisms are there? How are they shaped? How are they described? What are their general properties? So in qualitative terms, we would describe living entities. In fact, biology was first introduced as a reductionist science because it started off from botany and zoology, where people try to catalog animals and plants systematically. And that has therefore a very long tradition. And then later on, people try to look into those objects. They looked and tried to study individual organs, and then these organs were decomposed into parts and so forth. But still, this gives us also a qualitative view to biology, because we describe what we see and not so much how this interacts quantitatively. What I like here to introduce is the image of a mechanic clock. So if you think about a clock, it's composed of so many different gears that when you put them together, they show you the time, and that's a complex machinery, but every individual gear in that clock by itself doesn't do anything, but only if you put together the entire clock, then you will see what the time is. But to do that, you not only need a list of gears that you need, but you also need to know how they interact. And I think this is really a very good comparison, because the systemic view uses information from the reductionists in the sense of we need those lists of parts, but then we need to also have a detailed plan how these parts interact with each other. And that's what systems biology really is. So reductionism in molecular biology mostly implies a molecule-centric approach where we have individual molecules. So think about molecular biology. So the reductionist tries to identify every component of a system, but not their interplay. So, and these detailed lists are really important for us, but getting there is very difficult and takes a lot of time. So Professor Kohlbacher mentioned this a few times, that in the past, one postdoc or one PhD candidate was needed to describe a single gene or a single protein. And here at the bottom you see examples, 2 publications where this was done. So here the first publication covers one particular protein with a particular domain and what this domain does in Arabidopsis. Then the other example is about a specific kinase and a specific interaction that it has, but not in the greater picture of the cellular interaction. There are many more of this. So systems biology, and I mentioned that before, is an integrative approach. It takes the information from the reductionists one step further. So understanding what the components are and what they do is just the first step towards systems biology. But then we need their interaction. So, and only when we know what they do together and how they function together as a system, we will understand how the entire cellular system works. And that brings us to totally new properties, a systemic view to biology. And also at the same time, we need to simplify here and there because we cannot take every detail into account. There was once a saying that a perfect map of a city would be as big as the city, because then you can really cover every detail. That's not what we want. We need to be a bit more abstract. We need to reduce complexity so that our systemic view becomes simpler than the entire system. Otherwise, it doesn't make much sense. So, but then if you do that, it can reveal a lot of different properties from the system. So as an example, let's look at the circadian clock and its temporal dynamic behavior. And that is an emerging property and different from the interaction of the individual key players. So we see that here on that slide. So the circadian clock is in the simplest form found in cyanobacteria. And there you see a cycle of just 4 reactions. So you see the cycle starts here at the top with H1, and that H1 forms a hexamer. So H1 is just a monomer of a protein, and then that H6 is formed as a hexamer at a specific rate, and you see that that rate has the form of a sigmoid curve. And once you have that hexamer, that can bind a compound called B here with a rate K2, and that B binds to the hexamer at that rate, and then that H6B can bind with another rate that is characteristic, can bind a component A, and then the hexamer again disassembles, and we get the individual components back with another rate. And because every single process of these 4 processes here takes a certain amount of time, we can plot how this complex forms and what the concentration of that complex is over time, and we see an oscillating system. So it's a continuous oscillation where the complex is assembled and disassembled over time, over and over again. And what you see is these little square boxes in that plot. Those are measurements, and then the solid curve is the model, which is the result of a calculation from simulation. So the important part is really that we can construct a mathematical equation that describes what's happening here in the system, and then we can make a calculation and run a simulation that shows us the behavior, and we can, based on that, make predictions ahead of time, and can then tell experimentalists what plausible targets and experiments they could potentially do. So here, this slide now shows you some principles of systems biology, and that slide originates from a very big stakeholder in systems biology, the US Department of Energy. So why is the US Department of Energy so interested in systems biology? It's because what we really want is we want to go from the genotype to the phenotype. And once we can tell exactly what happens when we have a certain DNA, how the phenotype will look like of that cell, we could directly predict how we can make large amounts of interesting substances, for instance, long chain fatty acids that are very usable for producing a substitute of oil. As you know, we're running towards a crisis in which one day oil will be or gasoline will no longer be available, and then we might be we might need to have a substitute, and that can come from organisms that produce fatty acids on a higher rate for us. So you see, we're starting with a DNA, and then that gives us information about what proteins are in that cell, and those proteins give us information about the machinery, how they assemble and build complex enzyme structures, and that would tell us what possible reactions can take place and how the entire cell works. And once we know how a cell works, an individual cell, we can figure out how communities of cells work, how they operate jointly, what they consume or take up, or also secrete to the environment. And then we could make use of that by manipulating the genetic makeup of the cell. We could then create also organisms that clean up the environment by consuming a waste product that we want to get rid of, or to produce energy or precursors for energy, and so forth. Of course, also a big application would be building organisms or supporting organisms that help our microbiome to be more resistant against pathogens and so on. So these would be different use cases not shown here. So systems biology is really a genome-enabled science. This means that systems biology really arose when the first genomes were sequenced. And of course, there has been systems biology before. In fact, it is much older, and first biological models were already published in the 1960s and so. But the big advent of systems biology happened only after the first genomes were available. And here on the right, you see a circular genome for the first organism that was ever sequenced, which was Haemophilus influenza. And one person involved in the sequencing of first organisms is shown here, which is Craig Venter, and he is probably very well known to most of you. So what you also see on that slide is that the costs per genome sequence have significantly dropped since the beginning of this millennium, and they continue to drop, and they drop on a faster rate than Moore's law, which is very important in computer science and gives us the density of transistors in a computer. And that's already amazing how quickly that development happens, but the sequencing costs go down much more rapidly. And therefore we can expect that if sequencing organisms is so cheap, in the future we will have sequences more or less for everyone and everything. So which means also that if we could go from a genome sequence to the phenotype, we could really make patient-specific systems biology models and describe exactly processes that happen inside individual people, and then maybe also apply those techniques to design patient-specific treatments and strategies for treatment for healing diseases and so forth. So there's a lot of interest in here. But you also see there's a big gap. So computational power is growing slower then the sequencing costs drop, and therefore it's very important to have very smart algorithms for evaluating all these sequence data. At the same time, you see also on the right that the speed at which sequences can be obtained is going up, and that's going up very quickly. So within a very short time, we can nowadays sequence entire genomes, and that's really an amazing development with a lot of opportunities for us. So traditionally, when you think about a genome sequence, you would, as a bioinformatician, you would go through that sequence and try to annotate what is on that genome, and that would be a one-dimensional genome annotation. From top to bottom on that sequence, you would identify these different features such as translation, start site, coding sequences, genes, and so on. But systems biology goes beyond that. It's actually a two-dimensional genome annotation, which means that in addition to a list of components, you also try to encode the links, how they interact, these individual things. So for instance, you see that on that DNA, there are components encoded here. For instance, that circular item here, it's a gene product that has an effect on its own production or synthesis rate. Or here you see there are two different gene products that need to form a dimer, a heterodimer, to be able to catalyze a reaction of A to B. And that you see all reflected here in that matrix, at the two-dimensional genome annotation. So from top to bottom, you still have these different components that come from the DNA or that are floating around in the cell. And then from left to right, you say how they interact. So you see a -1 because in that reaction here on the left, A is being consumed, and there's a +1 because B is being formed. So in the same way, you can write down everything that is in the cell and how it interacts with each other. And because a matrix has these interesting properties that you can rearrange columns and rows, you can actually group things together that make sense in a certain way. For instance, at the bottom, we have here the chemical transformations. Then here we have the chemical transformation of compounds, or here we have chemical transformation of gene products, and here at the top we have regulatory interactions. So, and that's all in a matrix, and systems biology really comes down to creating such a huge matrix for interactions of items that are in a cell. And what you also see from here is we need the information from the reductionists to make such lists. And here omics data come into play. So what is omics? So omics, first of all, is a suffix to words, and not everyone is always happy with that, but it seems to be a very fashionable term. So omics data describes that we have an entirety of a certain object of a certain kind that we measure all together, and that's what is often called a neologism. So it informally refers to a field of study in biology, such as genomics, proteomics, metabolomics, but there are many more. So it started all with the genomics, from the genome as the entirety of all genes in an organism. But nowadays we have these different omics data types, so they all describe the entirety of a certain kind of things in the cell. So nowadays people like to make up a lot of different omics data types, and there are many that you might not have encountered yet. So such as the alignment tome or antibodiome and so forth. So if you're creative yourself, you can certainly make up another omics data type, but instead let's focus on those that are really relevant to us. So as you might remember, in systems biology, we want to go from the genome to the phenotype, and therefore we need a list of components in the cell and then want to describe the interactions. So now, first of all, let's make a list of different cellular components that are of interest to us. There's DNA, there's RNA, there's protein, there are metabolites, and then we have the fluxes. And the fluxes are the reactions, or the different speed of reactions, how much flows through cellular metabolism at a certain time. And now we have equipment that we can use for measuring these things. So to measure the DNA in a cell, we have a DNA sequencer. We can use RNA-seq to obtain information about RNA. With LCMS, we can obtain information about proteins. And then also we have this liquid chromatography mass spectrometry, or NMR, that we can use to also obtain information about metabolites. And these techniques you have covered already at the beginning of that lecture, but now there's a new data type here, the fluxomics, and here you will need to have radioactively labeled compounds, and then you have to measure how they disappear and how quickly products assemble, and then you can conclude how quickly the reactions operate. So what we really have is high throughput information. So we get DNA sequence data, we get mRNA profiles that are often displayed as sashimi plots. Then we have protein profiles here, we have metabolite profiles, and we have flux profiles, which are here displayed as a metabolic map. So, and those are the omics data types that we get: genome, transcriptome, proteome, metabolome, and fluxome, right? So the genome for the DNA, transcriptome for the RNA, protein for the protein in the cell, metabolome for all the metabolites that we find in the cell, and the fluxome for every metabolic flux in the cell. And now let's talk about the systems biology paradigm. Remember, from components we go to networks. From networks we go from computation to computational models, and those computational models predict cellular phenotypes. And now we have another omics data type here that is of interest for us. That's the so-called bibliome, right? So we take all these different omics data types, the genome, the transcriptome, proteome, metabolome, the fluxome, and the bibliome, and we integrate them all into what we call a knowledge base, or a reconstruction of a biochemical reaction network. And that's because everything that happens in the cell is essentially driven by the laws of chemics and physics, or in this sense, biochemical reactions. And therefore, if we know every biochemical reaction that takes place in the cell, we will know what that organism does. And that gives us a very valuable knowledge base, a collection of information, the big picture. So third, once we have that, we can apply a lot of different mathematical tools to that network, right? So from components, the omics data types, to one big cellular network, and then apply computational models here We can analyze the topology of that network. We can impose constraints on that network and see what happens to the system. We can analyze the dynamics of that network. We can perform sensitivity analyses. We can determine noise and run simulations in a stochastic framework. We can modularize that network and see how it's organized on a higher level. And we can apply gap filling, because every reconstruction will certainly have gaps, and then we need to figure out what we do with that, how we fill in information. So that's a science on its own. But okay, in the end, what we want to do is we want to go to the phenotype and application. So that means step 4 is validation, discovery, and use. And so what we see here is application. So we can look at high throughput data in a very new view and analyze them in a different, from a different perspective. We can use our models to discover missing parts, and we can understand biological processes very differently than by just looking at these individual parts of the system. And one thing that is of very big interest nowadays is designing biology or synthetic biology. So that's not possible without a good foundation in systems biology. So omics data are high throughput techniques. They provide us with a lot of different information. They give us different data types: genomics, transcriptomics, proteomics, metabolomics, or interactomics are listed here. So these are protein interaction data. We'll get to that also. So omics data themselves are reductionist data, but at a very large scale. They're often of very big volume, but the quality of individual measurement points is often not as good as in very detailed analyses. So when you compare classical data to omics data, you will see that the classical data are low throughput, whereas omics data are high throughput. Classical data often have a low dimensionality, single facts, whereas omics data can have a very high dimension, many different measurements and parameters in parallel. So the accuracy of classical data is very high. Every data point is supported by many experiments, whereas omics data often come with just one data point per measurement. Therefore, we have noise, randomness. So classical data are characterized by an analysis of experiments that is relatively easy, whereas omics data can often not be interpreted at all without statistical analysis or bioinformatics. So here you see a historic experiment type where an RNA sample is now prepared so biological object is being analyzed for RNA. So the cellular structure has been destroyed, and so everything is now in this little tube. So we have to amplify the RNA, for instance, using PCA experiments. And then in 2003, when this paper came out, it was very fashionable to hybridize that RNA with a microarray, and that microarray could then go into a specific reading machine, and that device here, and would give us a light intensity signal that tells us how much RNA is in a probe in comparison to a different probe. So we wouldn't obtain the exact amount of RNA that was in the cell, but we would be able to compare different conditions. So how much more or how much less RNA is in the cell under different conditions. And yeah, so that's how this was done. And here you see an experiment where this was done for a protein, so proteomics experiment, where a sample was directly used in a two-dimensional separation. So here we have the problem that we cannot amplify what is in the cell because there's no such technique such as polymerase chain reaction for proteins. Therefore, whatever is in that probe we can only use, and so we separate these different proteins based on two different characteristics. And so from top to bottom, you see here two different poles, negative and positively charged. And then from left to right, there could be the separation by mass, and therefore we get different spots on that two-dimensional jail, and then we can pick out the protein clusters that we have. But the problem again is that very low abundance proteins will be hard to detect. Also, proteins are very difficult to get out of the cellular membrane, and we have problems that effects such as glycolization change the properties of the proteins, and therefore they get more difficult to separate. But okay, so once we have that two-dimensional jail, we can pick out the proteins and then throw them into mass spectrometry analyses and then get information about what exactly we actually have. So that's also in the 2003 paper a technique that you might need to be aware of how this was done early on of that millennium. So okay, so once you have gathered information about the cell, you can look at the cell in terms of a network. And here is a network from the so-called KEGG database. That's the Kyoto Encyclopedia of Genes and Genome. It's a very popular database that tries to collect information about cellular components and how they interact. But KEGG isn't created to run simulations. They just want to catalog, like an encyclopedia, what happens. So it's actually a qualitative view to biology. And what you see is also that KEGG tries to always display the networks in the most generic form. Here you see now the citric, the TCA cycle for E. coli. And what you see is there are white and green boxes, and green means that an enzyme is present in E. coli, and then you see some white boxes, those are not present. So in the boxes labeled with EC numbers are the enzymes, and they can be white or green. So now let's focus on that enzyme here with EC number 2331, and that's catalyzing the reaction, the transformation of oxaloacetate to citrate. And here, when you click at that box, you get more information. So you see it's Escherichia coli K12, and here you see different pathways where that same enzyme is being involved. And you should be aware that those pathways listed here are conceptual entities that people made up to organize processes in the cell. In reality, there's of course not one pathway. Everything is connected in the cell, so all reactions are connected. Products of one reaction are the substrates of another reaction, and so forth. Therefore, there's no specific defined end or beginning of a certain pathway. But to display the information, it's very handy to organize information in that way. So of course you can also see much more information. So it has the genome sequence of the gene where that enzyme is encoded and so on. So on the bottom you see the link, so you can query that information yourself. Now let's take a look at one of these metabolites here, in this case citrate. So when you click at that compound, you will see what the structural formula is. You get different names for that compound, and again you see different pathway maps in which that compound is also involved. You get also information such as the sum formula or the exact mass and the molecular weight and so forth. So that's all of great interest, and many people who work in systems biology like to go to KEGG first for their analyses and to assemble information. So based on KEGG, you can really build a cellular network by connecting all these different reactions and extracting information from there. So that's very useful. Here's a different type of a network. So the KEGG network before was a molecular a metabolic network, and now we're coming to a protein-protein interaction network, and it's called protein-protein interaction because there are also other kinds of interactions that involve proteins, such as protein-RNA interaction or protein-DNA interaction and so forth. And now the question is, on the previous slide, we saw a metabolic network, and the arcs in that network were biochemical reactions. Now, what is an arc here in a protein-protein interaction network? Or to rephrase, what is actually a protein-protein interaction? So when proteins interact, that comes down to non-covalent interaction, such as van der Waals forces or hydrogen bonds formation between different proteins. And then you can see that there are many of those interactions in the cell. And it has been estimated that for human cells, there are even more than 600,000 different protein-protein interactions, where one molecule has a van der Waals interaction with another protein and so forth. So when you make a network out of that, it becomes what is often called a hairball, and it's very hard to analyze. Then a different kind of a network, signaling network or regulatory networks. And here you see on the right hand side what that is. Here's a display that you can often see in different publications, similar like this one here. So you see arrows between different objects, between different cellular objects. Here's one arrow pointing from what is called starvation, which is an external stimulus here, the starvation, that has some effect on adenosine monophosphate kinase, and that has another effect on other objects. But what you directly see from that figure is a very common problem in systems biology. Information is displayed, but it's not always entirely clear what is meant here. So we see that the same arrow points from starvation to the adenosine monophosphate kinase here on the top. The same arrow that also points from that kinase to the next object here in that chain of signaling molecules. But there's a different arrowhead here that points from REC1 to that other REC1 here. And it's not entirely clear from that drawing what that means. And also you see there are so many objects assembled at the bottom, and It might be a complex formation, but we don't exactly know what that means. And also we have color codes and different shapes. So that needs explanation. And what is really needed for systems biology to be a successful scientific discipline is one very clear, unambiguous display of information. And I will stop here and continue for the second half of the lecture after a short break.